Hi, I'm excited to be here with all of you and tell you about what we're building at Factorial Biotechnologies. I'm John Wells, the founder and CEO, and we're enabling full resolution NGS at scale. So the solution that we're developing over the last 15 or 20 years, there's been a lot of value created for patients and researchers performing what we call bulk NGS library prep, where you take a population of cells, you lyse them, and you purify the nucleic acids and sequence, and you get sort of an average of all the cells in the population. And it turns out that in order to really understand biology, you need to be able to characterize cells at single cell resolution to understand the differences between these cells. And what we're really excited to power with our early access customers are applications like understanding tumor heterogeneity and subclonal expansion under selective pressure, and also be able to characterize off-target edits at genome scale in CRISPR systems. And so how we're doing this is we're unifying single-cell NGS content and throughput. The last 8 years has seen tremendous progress in single-cell studies, but there has been a trade-off that's had to be made. You had to either pick high content with a very low number of cells or high throughput and more basic applications like counting applications and ATAC-seq. And so what we're coming into the market is to enable extremely high throughput, high content assays to be able to perform whole genome sequencing, full-length transcriptome, comprehensive genome profiling at the scale of tens of thousands of cells. And how we're able to do this is we've developed chemistries to perform massively parallel reactions inside of intact cells. So we can prepare complete NGS libraries inside of cells, massively parallel on all of the cells in a suspension simultaneously. And what this allows us to do is perform complex stacked reactions, but at an extreme scale. So not on one cell in a well, but on tens of thousands of cells simultaneously. And you can imagine it takes some special work with enzymes to prepare libraries inside of cells, and we're happy to be working with Ginkgo Enzyme Services to really realize the full potential of our platform. So it really is a very simple workflow to go from cell suspension to single-cell whole genome libraries in a single day. So you perform our in-cell library prep, and then you load this microfluidic chip that we have that our instrument can process 4 of them in parallel. Each chip can process up to 40,000 cells. You load the chip in a single stream and put it on the thermal cycler, and you have a library in a single day. And when I'm back up here, hopefully in a couple of years, what we'll see is that we've generated a significant amount of high content, single cell genomics data, applied advanced analytics and model training, and have new biomarkers and drug targets to show for it. So thank you very much.